we don't have much people studying vitamins and trace elements right now. Maybe for trace elements, zinc, copper, selenium, a little bit with iron. We still have a fair amount of people, but vitamins, there is none or almost nobody working on that. I could say maybe a handful of researchers working on that.
Hello, everyone. I'm Dan Columbus, your host for today's Swine Canada podcast. With me today, I have Dr. Danyel Dalto, who is a research scientist at the Agriculture and Agri-Food Canada Sherbrooke Research and Development Centre. What a mouthful. How are you today, Daniel?
I'm very good, and you, Dan?
I'm excellent. Thanks for joining us today. I think it'll be an interesting topic. So, Daniel, just because some of our audience may not be familiar with you and the research that you're doing, before we start talking about our topic today, do you want to maybe introduce yourself a little bit first, like where you've been and how you got to where you are?
Sure. First of all, it's a pleasure to be here and to talk to you. I'm sure it's gonna be fun. So as Dan just said, well, my name is Danyel Dalto. I'm a veterinarian from Brazil. And I did my master and my PhD in Brazil as well. If I try to translate it literally, it's Londrina State University in Brazil. I did my master in an area different from the one I'm currently working. I was studying intestinal health back there. But once I started my PhD, I was not studying vitamins and minerals as the area that I'm currently working on. I was once again working on intestinal health. But I was invited to come to Canada back in 2011. I was invited by Dr. Jacques Maj here from Agriculture Canada to do part of my PhD here with him in his lab. And it was a pretty new area of research to me that was vitamins and trace minerals to pigs, in pigs nutrition. And I decided to take this challenge. It was very new to me. I have never worked with these topics before, but I decided to come for the experience of living abroad and having this more how can I say that? A broad area of knowledge as well in swine nutrition. So I came to Canada back in 2011, and I did this animal trial here at the Sherbrooke Research and Development Centre. And in 2015, I finished my PhD, and I was offered a postdoc position. And I did this postdoc from 2015 to 2020 when I was fortunate enough to secure the position that I currently have as the lead scientist here for the swine nutrition lab at the Sherbrooke Research and Development Centre, focusing on the study of trace minerals and vitamins in swine nutrition.
Okay, great. I'm excited to talk about today's topic because I feel like trace minerals and vitamins is something that generally gets overlooked in the field of swine nutrition. That's because a lack of interest, or it's just the difficulty potentially in doing some of this work. So I think it'll be interesting.
I would say it may be both of your points. Because if we go back in time, I would say back in 1920 about, when people started really looking into vitamins and trace minerals, what they discovered back there is that we needed these nutrients to avoid deficiency in pigs and other species. However, once these deficiencies were completely or mostly disappeared from animal production, of course, we tried to start using higher levels of these vitamins and trace minerals to improve performance, for example. But when we use vitamins, especially for minerals, we have some exceptions. For example, zinc and copper. But for most of the other minerals, and especially for vitamins, when you increase levels in the diet, you won't see a great improvement in performance. That's not what vitamins and trace minerals, most of them, do. These elements, they work mostly on health aspects. So it improves the metabolic health. That's like the way I like to call it. It's the metabolic health. We work on immune system. We work on antioxidant system. We can modify or modulate microbiota. But growth performance, that's not the focus. But back there, that was the focus. So once they realized that we were not able to improve growth performance with these nutrients, I think the scientists lost the interest in studying these nutrients. So we don't have much people studying vitamins and trace elements right now. Maybe for trace elements, zinc, copper, selenium, a little bit with iron, we still have a fair amount of people, but vitamins, there is none or almost nobody who working on that. I could say maybe a handful of researchers working on that. Of course, there are difficulties, analytical difficulties, and things that in the trials there we have to take care of special kinds of sampling tubes, and the timing between sampling and the processing of samples. There are a lot of particularities that we have to take care of, but maybe these are still a little hard to work on because we don't have much improvements in terms of the study or the understanding of these elements right now.
Mm-hmm. Hopefully with more interest in it, then we start to develop those a little bit better. It's interesting you talk about the focus on growth is what basically kind of, led to a limitation in this type of research, because we're seeing that in other nutrients as well, right? We're with amino acids, it's like we're seeing that, they have benefits beyond just growth, but if you just measure growth performance, you don't see anything and you think that there's no point, right? So it's interesting to see that it's in multiple areas, but then this focus on health that's come out in the last couple of years has really started to show more of what nutrition can do and what other nutrients can do.
Yeah, and the study with vitamins, for example, you mentioned amino acids. Well, the study of vitamins, we can do a lot of stuff playing with amino acids as well, because many vitamins, for example, vitamin B6, it has actions in the metabolism of a lot of proteins and amino acids. So we can try to improve the efficiency of utilization of other nutrients by studying or using different vitamins, for example.
Yeah. So I know you've developed work in kind of three main areas, and your research is focused on kind of these areas, one being reproductive biology, pre and post-weaning metabolism, and sustainability. So I think, we'll just work through those three areas and kind of your major findings and kind of and thoughts of those. So we'll start with reproductive biology, right? So how about you just discuss maybe some of the stuff that you've done in that area?
Yeah, I started studying at this area back in my PhD when I was working with the gestating cells. We were testing different sources and levels of selenium interacting with vitamin B6 in the diet of gestating cells, trying to evaluate if these nutrients could have any kind of impact in terms of cells reproductive performance, but also or especially on embryos development. Because as selenium has antioxidant effects, and that are very interesting. We believed at that time that the ovulation process that is known to produce a lot of free radicals that we call ROS, reactive oxygen species. So we know that ovulation produces a lot of ROS. But as it's a local production, we were not sure if it would have a systemic impact in the soil. And we started evaluating this aspect. So we were trying to prove that the ovulation or the peri-estrus period would be an interesting experimental model for oxidative stress. And our results showed that although this is a local process, the antioxidant stress is a systemic, has a systemic impact in the soil. So with this knowledge, we started using selenium with its antioxidant effects to try to reduce this systemic oxidative stress that the soil will go through during the ovulatory process. And in a first study, we were able to show that we are indeed able to reduce the oxidative stress, the systemic oxidative stress, and also improve some parameters related to not only embryo development, but also litter size. So we were able to increase the number of, how can I say that in English, yellow corpse or hemorrhagic corpse that will be the corpus luteum some days later. So it's an indication that we had more embryos or more follicles that were ovulated from that cell. And we were also able to show an increase in the peak of LH, of luteinizing hormone, in the cells, once again showing that the cell was producing more of this hormone that is so important for ovulation. In a second study, going a little further, instead of studying ovulation itself, we went to the 5 days embryo and also to the 30-day embryo. And we were able to show that at 5 days of gestation, the combination of selenium, organic selenium actually, and vitamin B6 improved or increased the number of more mature embryos at 5 days of gestation. And that in the absence of degenerated embryos that were much higher in the inorganic selenium treatment and also in the control treatment. Between control and the inorganic one, we didn't have any difference. But organic selenium improved a lot the development and the quality of these embryos. And these results were reproduced at 30 days of gestation, where these embryos were heavier than the ones in the other two treatments. But we were not able to increase the amount of antioxidants in the embryo. So we were not understanding why we feed animals with selenium compared, for example, to the control diet. We can enrich these animals, these embryos, with selenium. So selenium is being transferred to the embryo, but there is no change in terms of the antioxidant capacity of these embryos, at least in terms of selenium-dependent antioxidants. So by going a little further in these studies, we found out that part of the metabolic pathway that goes from organic selenium or selenomethionine to the synthesis of seleno enzymes, this metabolic pathway is not complete. It's not mature enough in the porcine embryo. Actually, this metabolic pathway will be complete some weeks after birth. So even by the end of gestation, there is nothing happening in terms of seleno enzymes being produced in the embryo from selenomethionine. So what it means is that the embryo receives or gets selenium from its mother, but it doesn't come, but it's, how can I say that? He's receiving selenium as organic selenium because whatever we feed the cell, if it's organic selenium or inorganic selenium, this molecule will bind any kind of organic matter, whatever, if it's amino acids or other enzymes within the body of the mother. And this organic molecule will be transferred to the embryo. But as we can have a greater transfer of organic selenium, for example, selenomethionine, that will enrich the embryo, but we are not able to synthesize enzymes, it leads us to think that some other complexes between selenium and I don't know which other molecule would also be transferred to the embryo in a more metabolized way, closer to what is needed to synthesize selenoenzymes. So by the end of the day, what matters if we want to improve embryos' antioxidant capacity, it's not the source of selenium that we are feeding the mother, but the amount of selenium that we are feeding the mother. We have to improve as much as possible selenium concentration in the cells, so we will transfer these selenoproteins or these proteins that are binding selenium in some way that will be transferred to the embryo. But we still don't know what is the effect of this kind of supplementation when at birth. We don't know if these differences, these significant differences that we were able to create between organic selenium treated cells and control or inorganic selenium treated cells, we don't know if these differences will remain significant until furrowing, for example. So this would be something that we are interested in study.
Yeah, I was going to ask if the development of those systems happens after birth and you have these enriched embryos, right? Like, does that help with that development after? But it sounds like that's something to come.
Yeah. It's something to come, and I'm trying to work on that. But one other interesting aspect is that if we consider the antioxidant system, we have at a first step in the system, we have SOD, superoxide dismutase. A second step will be the metabolism by glutathione peroxidase, that is seleno-dependent. And then we have kind of complementary systems, for example, catalase and others. So if we are not able to improve glutathione peroxidase levels in the embryo because of these particular aspects of selenium, maybe we can work with SOD. And SOD, it's dependent on zinc, copper, and manganese. So maybe the supplementation of these one or the three of them, the three of these trace minerals in cells during the beginning of gestation or even before working on once again ovulation, it may be a very interesting approach that should be further studied.
Yeah, it's interesting. You mentioned that it's not the source of selenium, it's the amount that you feed the cells, but you saw a difference between the organic and the inorganic selenium. So what is I guess doesn't that indicate that source would be important as well?
I would say that we have to consider what is the objective of this supplementation, because as I said, it's not the source of selenium when we think about embryo antioxidant system. But if we think on the whole system, mother and embryo, everybody together, organic selenium seems to be more interesting because it has two pathways during its metabolism. It can go to the deposition in tissues, for example, in proteins, and it can go to the synthesis of antioxidant enzymes. So what is interesting about organic selenium is that the difference between these two metabolic pathways, how the metabolism will decide when selenium goes to protein deposition or when it goes to antioxidant enzymes synthesis. It's regulated by the level of oxidative stress. So I say it's interesting because in inorganic selenium-treated animals, basically all selenium will go to the synthesis of selenoenzymes, whatever if the animal needs it or not. So we can say it's a little it's kind of wasting a little bit of the selenium, because you don't need antioxidants and you are producing it a lot. But with organic selenium, it will be produced when the animal needs it. So it's like a feedback approach, where when levels of oxidative stress increase, the level of selenium enzymes will increase as well when you feed organic selenium. If you don't have oxidative stress or a basal level of oxidative stress, this selenium will be directed to the deposition into proteins. And that's what we observed with our results, whatever if it's on the activity of these antioxidant enzymes, or in the gene expression of these particular genes. So that's the difference. When you talk about cells, organic selenium is very interesting. When you talk about embryo, we have, embryo antioxidant system, there's the selenium source is not that interesting, but when you talk about embryo development, it was very interesting. So overall, I would say that organic selenium, it's a better choice.
Yeah. Okay. So you've also done some work with trace elements in litter homogeneity. So what have you found with that?
Yeah. Actually, the studies that we've done for pigs homogeneity, it's the ones with selenium as well. But instead of looking this homogeneity at birth, we were studying with embryos at 30 days of gestation, and that's where we were not able to find any difference in terms of this homogeneity at 30 days of gestation, whatever the level or the source of selenium. And that was a little disappointing. But as I said, maybe if we go up to favoring these selenium effects, they may be increased over time. As we saw at 5 days of gestation, a better development. At 30 days of gestation, a better development. Maybe at favoring, this will be even potentialized. But it has to be studied yet.
Okay, well, we'll look forward to seeing that. Everybody's always interested in the long-term effects, right? So it's the problem we all have. You show it in the short term, and then everybody asks, well, what about in the future? So the second theme of your research has done these trace elements in what was it, the pre and post weaning metabolic development. So obviously after in the piglets. So what? Maybe let's talk about that a bit.
Sure. We've done some interesting projects here, studying the bioavailability of some trace elements. We've worked with zinc, copper, iron, and selenium. And for zinc and copper especially, we were looking at levels and sources, and also the ratio between zinc and copper, which is very important. And the main finds that we had is that for zinc, the source is an important aspect. So organic zinc is more, I don't know if I can say bioavailable, because the approach that we have here is a very particular one. We don't work with digestibility, for example. We work with an approach that we call net portal appearance. What we do is that we perform surgeries here, where we install a catheter in the portal vein. Just to refresh our memory in terms of anatomy, the portal vein is a single vessel that drains all the blood coming from the gastrointestinal tract. So everything that is absorbed from the stomach until the rectum, basically, will necessarily go through this portal vein. So that's why we cannulate this vessel. So we install this catheter at the very entrance of the liver to be sure that we are collecting as much blood as possible. We don't want to lose not even the blood coming from the stomach. We don't know if there is absorption of trace elements in the stomach, but we don't want to lose them if they exist. And we install a flow meter around the portal vein as well, very close to this catheter, to measure the amount of blood going through the vein per minute or per hour, whatever. And then we install a second catheter in the carotid artery. It could be any artery because based on some tests that we've done here, venous blood will change considering the organ that it's draining, but the arterial blood is basically the same whatever you take samples in the organism. And the explanation is very easy. Well, when it goes into the heart, it's mixed, and so there's no difference between that. So we take the carotid artery because it's easy to access. And after the surgery, what we can do is to take blood samples in both vessels at the same time. So we have the portal blood and the arterial blood. And by doing a simple subtraction between portal blood and arterial blood, we'll have the net concentration of the nutrient we are studying. For example, why we have this net concentration? Because if we think that all blood going to the portal vein comes from the intestinal tissue, for example, and the intestinal tissue, the input comes from diet, but also from the systemic blood that's nourishing these cells. So in the portal vein, you have the mix between nutrients coming from the diet after absorption from the intestinal tissue, and the intestinal tissue will release these nutrients in the portal vein, but also part of these nutrients coming from the systemic circulation. So you have to subtract what is coming from the systemic circulation to have an idea to better estimate what was really absorbed by the animal. But it's not absorption itself, because we have to consider that the intestinal tissue will use some of these nutrients. And that's the beauty of this technique, because we cannot estimate how much is used by the intestinal tissue, but we know how much is released from the intestinal tissue to be used, metabolized by the liver. So that's why I don't know if I can say it's bioavailable, but it's the net or the real absorbed or the real available amount of nutrient that the animal will have to use. So we've done that with the zinc, copper, and as I said, for organic zinc, it has a better absorption or better net availability of zinc. Whatever the level we feed the animals, if it's higher levels or lower levels, the organic source is always better than the inorganic one. And even within the inorganic sources, we have those that are more bioavailable or more available. For example, zinc sulfate. It's not in the literature, but this technique was precise enough to show that we have basically twice the availability of zinc coming from zinc sulfate compared to zinc oxide. And as much zinc we feed the animal, as lower is the proportion of this zinc that will be absorbed. And also we were able to show that zinc absorption happens 90% between the first hour after a meal. So within 1 hour, we have a very beautiful peak of zinc in the blood, and then it goes to basically basal levels. For copper, on the other hand, nothing happens. There is no effect of levels. There's no effect of sources. There's no effect of feeding the animal. You feed the animal, and during the following 10 hours, serum copper's level don't change. So it's kind of weird, because you feed a lot of copper to the animal, so you are expecting to have a peak of copper in the serum, but it doesn't happen. Why? We have other experiments that we may talk about later that will explain that. But basically, most of this copper is retained, is trapped in the intestinal tissue. But we can try to explain it later. But what is very interesting in these projects, in these experiments, was that in the treatments where high levels of zinc and copper were added together, we had this combination of high zinc and high copper. This net availability of copper was actually negative. And what it means, a negative availability, it means that not only dietary copper was not present in the portal vein, but maybe some of the systemic copper was also retained within the intestinal tissue. So the intestinal tissue was, both using or trapping all copper that was going through it. And that gave us a negative value for this availability. So we were interested in understanding a little more what was happening with that. So we did some other studies, more long-term studies, evaluating not only the level of zinc and copper in different organs, for example, the intestinal tissue, the liver, the kidney, and also the serum, blood serum, but we have also done some PCRs to better understand what was happening in terms of transporters, so zinc and copper transporters in these different organs. And what we found was an explanation for all that that we saw with the bioavailability studies. Actually, when we increased the amount of zinc, and we studied 3 different amounts of zinc, 100, 1000, and 3000 ppm of zinc, as much zinc you feed the animal, as lower will be the expression of the transporter responsible for absorbing zinc from the intestinal lumen. So this is a homeostatic mechanism where the animal is trying to avoid absorption at the first place. However, it's not difficult to understand that it's not if the proportion of zinc is reduced, the proportion of absorbed zinc is reduced. It doesn't necessarily mean that the whole amount or the total amount of zinc will be reduced as well. For example, if we consider that animals receiving 100 ppm of zinc will have a 20% absorption of this zinc, we have 20 milligrams of zinc that will be absorbed. But if we take an animal and feed them 3000 ppm of zinc and we consider a 5% absorption, it's still higher than the 20 milligrams of the one fed 100 ppm. So it's a first step in the homeostatic control of zinc, of body zinc, but it's not efficient enough to really avoid any kind of toxicity. So a second step is the release of zinc from the intestinal tissue to the portal circulation, for example. And it's interesting because the transporter doing this job, it's more expressed when we increase the levels of zinc, meaning that the intestinal tissue is trying to avoid its own toxicity. So it reduces absorption, but it increases the release at the same time to reduce as much as possible its own levels of intracellular zinc. As a consequence, in the liver, we have an increase in the absorption of zinc, the concentration of zinc in the liver, but also in the blood serum and in the kidney. So the animal, when we go from 100 to 1000 ppm, the animal is able or efficient in regulating these zinc levels. But when we go from 1000 to 3000, this regulation is completely lost. The animal is not able to handle that much of zinc in its body. And what is the effect on copper metabolism, for example? On copper metabolism, what is happening is that zinc has no effect on the absorption of copper, or apparently don't have any effect on the absorption of copper itself. It doesn't play any role in increasing or reducing the expression of the gene responsible for this absorption of copper. Neither for the intracellular transport of copper, neither for the release of copper from the intestinal tissue. Neither in the liver and neither in the kidney, basically, or very low in the kidney. However, there is one single and very important intracellular metalloenzyme called ferritin. Oh, not ferritin, sorry, metallothionein, that has a very particular response to zinc. It increases when you feed animals higher levels of zinc, so it's responsive, very responsive to zinc. However, it has a much greater affinity for copper, which means that when you feed high levels of zinc, you increase the synthesis of this metal enzyme, but it's gonna bind a lot of copper, not zinc, copper. And what is the first tissue where copper will get in? Zinc and copper will get in after ingestion in the diet. It's the intestinal tissue. So most of this dietary copper, and we can think on about the systemic copper as well that is coming to the intestinal tissue to nourish the cells. Most of this copper will be trapped, will be retained within the intestinal tissue, and will not be released to reach the liver. And that's what we see in these studies. We have a very enriched intestinal tissue with copper, and liver levels decrease a lot. I would say that it decreases to levels we see in piglets at birth. So it's kind of a 2 to 3-fold decrease in liver copper concentrations when we feed 3,000 ppm of zinc to animals compared to those feeding 100 ppm of copper. But what else? But how can I say that? One other step in this metabolic control, or this metabolic imbalance that zinc causes on copper metabolism, happens in the kidney, because in the kidney, this metallothionein, this enzyme that is responsive to zinc, will increase as well, because the liver is enriched by zinc. So it increases the synthesis of metallothionein. But what is very particular in the kidney metabolism is that we have not only the intake of a nutrient, whatever nutrient, but we have filtration and the reabsorption of this nutrient. So in the kidney, metallothionein will have twice the chances to bind copper as it had in any other organs. So the levels of copper in the kidney increases a lot as well. So we can see that in the intestinal tissue where we have absorption, we have high levels of copper. In the kidney tissue where we have reabsorption, we have a very high levels of copper. But in the serum and in the liver, that's where copper is needed to be metabolized and to be transported to the other organs. We have very low levels of copper. So the conclusion of these first studies were that when we increased levels of zinc in the diet, we may create copper deficiency in the post-weaning piglets, which was at least not known before these studies.
I mean, that's fascinating to me. It obviously has some pretty big implications for those nursery diets, and especially when we're looking at zinc and copper as, non-antibiotic pathogen control mechanisms, right? That what are the negative consequences that we're having on those pigs as a result of that?
Yeah. And then after that, we were interested in evaluating the different ratios between these two elements. What happens if we reduce zinc levels and we increase copper levels? Does copper has the same negative effects on zinc metabolism? So we started with this that same approach with the portal vein, the surgeries and the portal vein approach. And basically what we observed is that when we work with lower ratios between zinc and copper, the metabolism or the absorption, the net availability of both of them will be better. But once again, when we goes from a lower level of zinc to a higher level of zinc, in this ratio, we keep the same, even if we increase the levels of copper, the increase of zinc will, how can I say it, that overcome this higher levels of copper, and we will have negative values once again. So the right way to face this issue is not increasing copper levels, it's really decreasing zinc levels. But a very interesting result we got is that when you increase copper levels, you increase zinc absorption or zinc availability. It may be related to the metallothionein enzyme, because if we think that metallothionein will bind copper, and by binding copper, it will leave more zinc available, more free zinc to be released by the intestinal tissue. So we have this effect of copper in improving the. We were expecting maybe to decrease, to impair zinc absorption or zinc availability, but at the end of the day, copper supplementation or higher levels of copper in the diet will actually increase zinc absorption or zinc availability to the animal. And then with these results in hand, we performed a second study, a long-term study, looking at those same parameters that we mentioned before, levels of zinc and copper in different organs, gene expression in different organs. And with this approach of high and low levels of zinc, high and low levels of copper, we were able to have better insights in terms of copper metabolism. In the first study, where we variated only the levels of zinc, we were more focused on zinc metabolism. We had a lot of information on copper metabolism, but our focus was really zinc metabolism. But in the second one, as we changed a little bit the levels, we worked with different levels of copper. We were able to look into how copper metabolism reacts when you have different levels of zinc, but also if it can counteract those high levels of zinc with higher levels of copper. And the answer is, once again, as in the availability study, the answer is no. Whatever, if you have low or high levels of copper, the negative effects of zinc will remain there and very significant. Of course, that when you have low levels of copper, these effects are even more significant. Less the copper will reach the liver than if you have more copper being fed to the animal. But even there, you have a reduction in copper levels in the liver, and the animal can become deficient at a given moment.
It's never as easy as you think it is. But I love that when you start looking into things, you see these kind of unexpected effects, right? So I guess that's a good segue into your final theme, especially talking about zinc and copper, because you do a lot or have done work on sustainability and limiting environmental impact. So I think this is something I think it's been on people's minds, especially with zinc, and we know what's happening in the EU with their limits on zinc and that. So I guess we'll our last topic will kind of be on that side of it.
Yeah, sure. And if I can add just something that you talked about, you mentioned about environmental impact. Of course, these studies were not focused on the environmental impact of zinc and copper, even if we were studying pharmacological levels of zinc and copper. But if we think that copper is trapped in the intestinal tissue, and the intestinal tissue, the turnover of intestinal cells is 25% per day. Within 4 days, you have 100% renewal in your intestinal cells. All this copper that is trapped in the intestinal tissue will be lost, and you will find it in the manure. So even if it's a nutritional study without any look at the environmental aspect, you can have something. You can keep in mind that you are losing a lot of zinc, you are losing a lot of copper by doing this kind of supplementation. Of course, I'm talking about nutrition. I'm not talking about control of diarrhea, for example. We know that high levels of zinc oxide they are efficient in these aspects, but nutritionally speaking, it's not that interesting. and even for animal, and even for animals' health, because we can create copper deficiencies. And I didn't talk about iron, because we evaluated iron metabolism as well in these studies. And although the effect of zinc, of high levels of zinc, are not that intense as we observed in copper metabolism, we do have effects on iron metabolism as well, and these effects goes in the same direction as we observed for copper. So when you increase the levels of zinc, you increase the levels of a very particular enzyme in the cells called ferritin, and this ferritin will bind iron. So it's basically the same approach. You increase zinc, you increase ferritin, you bind iron, you will trap iron in the intestinal tissue. You don't have enough iron reaching the liver. But it's not that bad as it's for copper. So for iron, what I could say is that high levels of zinc in the diet will not create a deficiency of iron, will not create or stimulate the development of anemia, but it will decrease the capacity of the animal to build up iron reserves at the long term.
I think it's fascinating. I'm glad that you're over there doing this work. It's out of my wheelhouse, but I'm glad somebody's working on it and looking at these things. And I think this is great. We are running a little bit short on time. So before we get to our final three questions that we asked everybody, I like to, provide you the opportunity, like what's one take-home message or something that you hope that the listeners of this episode will get.
Yeah, well, for sure that in terms of if I pick this this main theme that I tried to bring here, that is zinc and copper, we know that in Europe we already have some restrictions in terms of zinc supplementation, pharmacological levels of zinc, and it's coming to Canada. I don't think it's gonna take that much of time before we are not able or not allowed to use these kinds of strategies in our herds here. So we still don't have, and maybe we won't have that silver bullet to replace zinc oxide. So of course a more holistic approach is needed. But to think on a holistic approach, we do have to understand what is happening with trace elements. Here I'm studying zinc, copper, iron, selenium, but what is happening with vitamins? We know that zinc may interfere with the metabolism of other metals, but copper can do the same. Manganese can do the same. So we need... I don't like to say urgently, but it's kind of, we need more research, we need more understanding on these metabolic aspects. We cannot look at only on growth performance. Of course, in terms of money in the pocket of producer, growth performance is important. It's imperative for the industry, but to keep having good growth performance, we have to look at more basic stuff as well. Okay, so we have to go deeper in the metabolism. We have to go deeper in the physiology, because by understanding that, we'll be able to have better products that will be more focused on a particular problem or a particular issue. So for zinc and copper, we can work with lower levels. Yes, we can work with lower levels. We need this holistic approach. Of course, we have to work on the sanitary conditions of pigs, the environmental conditions of pigs, welfare conditions of pigs, nutritional conditions of pigs. But we have to know the basics. So we still don't know what are the real requirements of trace elements in pigs, trace minerals and vitamins. All our or most of our work that we see in the official recommendation tables, they're quite old. They were done in the 1980s and sometimes even before. So we have to have a much better understanding of these aspects if we want to have more robust piglets, piglets that will not get sick that often or that easy, and if they get sick, they can recover faster and better.
Yeah. I mean, we said that right at the beginning, right? That they're old and they're also based on growth. And I think that's one of the things when it comes to a lot of the nutrition research not moving forward is that we have to focus on more than just the growth if we really want to solve this problem and really and find that, that solution to reducing antibiotics and making a more robust pig. So I completely agree. So we end each podcast with 3 questions that we ask everybody that are kind of beyond the research thing, but potentially of interest to our viewers, or listeners, I guess. The first is, what is your favorite swine-related book or resource?
My God, it's interesting because I'm a nutritionist, but the one I like the most is the one I have here. It's called Disease of Swine. I'm really passionate about pathology and I'm a veterinarian, so I think it's a natural thing. And I tried to bring a little bit of this veterinarian expertise in my work as a nutritionist. So that's where I try to do some experiments with challenges, if it means something to you.
Not only that, I think that's good. If every nutritionist came out here and said NRC, it would get really boring pretty quickly. So it's nice to hear different resources. So our second question is then, what is your favorite book or resource outside of agriculture? And this could be, fiction, nonfiction, anything. Something that you're reading now or have read.
Well, what I'm reading now, well, I'm not a very enthusiastic reader, to tell you the truth. What I do when I'm not working and taking care of my 2 kids that consume a lot of my time, but I really like to work with flowers. So I go from animals to plants, and that's what I try to relax a little bit.
Yep, that works too, as long as there's something.
Well, of course, here in Canada, it's a little restricted. I cannot work on that like 12 months of the year as I did back in Brazil. It's here we have less time to work on the soil, but it's okay.
A little bit shorter.
Yeah, a little bit shorter.
Yeah. So, and so our final question is, in your opinion, and you can just define success however you want for this question. What sets successful swine professionals apart from those who are not?
Well, I would say that a successful swine professional is someone that may have some kind of expertise in a particular field of the production or whatever, if it's nutrition and nutrition, health, welfare, animal welfare. But it's someone that have a more general knowledge of the system as a whole. Because sometimes we get so specialized in gestating cells, in lactation cells, in post-weaning piglets, but we have to keep in mind that it's a system that goes from even before birth, from the cells in the reproduction herds to slaughter, and even after slaughter, with the assurance of the quality of the final product. So we have to keep in mind that it's a whole food system, it's a whole production system, and that we have to understand. We don't have to be a specialist in the whole thing, but we have to understand the whole thing. And by doing so, you will be able to work better in your specific field. You can do very good stuff working on cells reproduction if you keep in mind that those piglets born from that cell will be a meat at a given time. So you have to think about it.
Yeah, I agree. I think it's something that we tend to all be guilty of at some point, right? Where we get so focused on what we like to do and what we do. But yeah, understanding where you fit in that big picture, I would agree that it's a great thought. Well, I think that's bringing us to the end of our podcast. So I'll thank everybody for listening, and obviously thank you for coming on and talking about your work. I think it's great, and looking forward to seeing what comes out. Maybe have you on in the future for some new stuff.
Well, sure. And as I said, it's my pleasure to be here, and I'll be glad to be here once again if you invite me. So thank you.
I'm sure we'll have you on in the future.
Had a good.
Yeah. Thank you.